Good morning, good day, and good evening, wherever you are in the world tuning in to the first of our Antibody Discovery webinar series. Thanks for joining in and taking the time to listen in. My name is Vincent Pai, and I'm incredibly excited to kick off this series with this in-depth plunge into antibody discovery with a longtime partner, Twist Biopharma Solutions, our division of Twist Bioscience. I'm joined today by Colby Soders, CSO of Twist Biopharma Solutions, who you'll hear from more in his presentation titled A Meta-Analysis of 150 plus Antibody Discovery Campaigns, Transgenic Mouse Performance Against Single B-Cell Technology Trends. Before we jump into today's discussion, I first wanted to cover a few notes about our webinar series. I'm extremely grateful for our partners, Twist and Genovac, for helping to organize what I believe to be an exciting and interesting series for the webinars, starting with today's presentation and continuing through the next 2 weeks. If you haven't already registered for our session with Genovac and/or a roundtable between myself, Colby, and Brian, the CEO of Genovac, head over to our website at Bruker Cellular Analysis to sign up. Thanks, and looking forward to seeing you all there. And now, on to today's discussion. Before we dive into Colby's presentation today, I wanted to give a brief overview about Bruker Cellular Analysis and talk a little bit about the optofluidic platform used for antibody discovery. Bruker Cellular Analysis is a part of Bruker's highly innovative and technology-forward instrumentation, and we provide scientific leaders and innovative researchers access to 2 single-cell functional multi-omic platforms: the IsoSpark line of systems and products for bulk and single-cell proteomics, and the single-cell optofluidic platform and products for antibody discovery, cell line development, and T-cell profiling. These 2 platforms are at the forefront of Bruker's strategic entry into single-cell biology and the multi-omic space, and both technologies continue to provide innovations across a range of life science applications. We just finished 2 great webinar series discussing our cell line development and T-cell profiling applications. So if you're interested in learning more about those, head over to our website to enjoy those webinars on demand, of course, after this one today. Today, however, we'll be focusing on the Beacon optofluidic platform and our antibody discovery application, which we enable rapid, species-agnostic, function-first single B-cell screening, powering monoclonal antibody discovery for therapeutic, diagnostic, reagent, and research end uses. There are 2 key technical innovations that the Beacon platform hinges on that serve as the foundation for all of our powerful applications. The first is the horizontal nanopen and channel design, which exploits tiny volumes and laminar flow of our microfluidics to enable real-time measurement of secreted molecules, cell-cell interactions, and diffusion-led culture for up to 28 days, all without perturbing the cells and particles inside the nanopen chambers. The second innovation is with optoelectropositioning technology, which enables manipulation of single cells or beads with our patterned light, enabling the finest control in creating single cell or even multi-cell experiment conditions at massive parallel scale. By leveraging these 2 key innovations specifically for the antibody discovery application, we've created several products and workflows as part of what we call the OptoB discovery portfolio. We start with the OptoB discovery workflow that starts with an immunized sample, whether from mouse, rabbits, camelids, including alpacas and llamas, or even human donors. Bruker Cellular Analysis provides reagents and know-how to isolate and, if needed, activate B cells into antibody-secreting cells from those samples. These antibody-secreting B cells, whether they're primary plasma cells or differentiated memory B cells, are then loaded onto the Beacon system into our OptoSelect chips. Once the B cells are positioned with a single cell per nanopen chamber, assays are performed quickly to assess a variety of different phenotypic and functional characteristics of the secreted antibodies, which all layer together and inform hit selection. Hits can then be recovered into well plates using a number of different methods. And further downstream molecular biology processing is supported by our products and reagents to prepare libraries for sequencing. Depending on the sample and sequencing that is used, this whole process can take a single week, with the on-beacon screening typically taking just a single day. The OptiSelect 11K and OptiSelect 20K chips are regularly used for antibody discovery on the Beacon platform, which offer the highest throughput of 11,000 or 20,000 nanopen chambers, respectively. When using OEP to load cells at massively parallel scale, users often see about 50% to 75% penning efficiency, resulting in up to 60,000 single B cells loaded across four 20K chips. Here, we see single cells being loaded in a single field of view, just one of many, in massively parallel throughput, leveraging our fully automated cell detection, ideal path calculation, and management of our OEP cages. Once the single B cells have been loaded into individual nanopen chambers, users can immediately observe the secretion of antibodies from these B cells in real time due to the miniaturization of the nanopen chambers. As the concentration of these secreted antibodies increases, they diffuse outwards from the nanopen chambers into the main channel. It is here where we leverage the fluidic control to bring different assay reagents into the main channel, park them above, or even move them into the nanopen chambers and allow diffusion to take place to assess the antibody specificity and or function. Especially for assays that are performed in the main channel, reagents can simply be flushed out of the chip to reset the chip, allowing additional assays to then be run in sequential order by importing new reagents, enabling comprehensive screening for multiple antibody characteristics for each antibody on the chip. In this way, we can enable real-time assessment of antibody characteristics as quickly as 20 minutes, as shown on the right by a multiplex antigen specificity assay. By adjusting the different assay reagents that we park in the main channel and or inside the pens with the antibody secreting B cells, we can assess a variety of different antibody binding specificities, cross-reactivities, binding affinities, and even functional characteristics like blocking, internalization, and agonism, both for soluble antigens as well as more difficult membrane-bound antigens. These assays can be mixed and matched, enabling multiple readouts of the same kind. Say, if you're looking for antibodies that cross-react to mouse, non-human primate, and human homologs of the same antigen, or combining those with other assays like internalization for unparalleled flexibility and modularity for every workflow, target, or end application. After performing the single or multiple serial function first assays, recovering BCR sequences from selected hits occurs in a fully automated user-defined process. The simplest is to just leverage OEP technology, what we call cell unload, to move a single cell back into the main channel where we dispense the cell into a single well of a well plate to maintain heavy chain light chain clinality. Another technology that we can leverage is also called the OptoSeq BCR unload, which allows us to co-locate mRNA beads into pens of hits of interest. We lyse the cells on the chip, rupturing all the cells in the system, and releasing mRNA from B cells of interest to be captured by those proximal mRNA capture beads. And subsequent reverse transcription occurs on chip to stabilize those mRNA transcripts into cDNA. Finally, of course, we can then recover clonally those mRNA capture beads that contain the cDNA one at a time for downstream processing to prevent mRNA degradation and overcome limitations of BCR recovery. Of course, then we provide a lot of protocols and know-how to perform library preparation for BCR sequencing, typically with Sanger or even for NGS. Taken together, the Beacon platform for antibody discovery enables researchers to join functional assay data to the BCR sequences at unparalleled depth and scale, which not only leads to better informed lead candidate selection, but also for those interested in developing or leveraging in silico AI lead optimization tools, broader context around the antibody design space, even from those less desirable antibody sequences that you've discovered. True to Bruker's motto of innovation with integrity, Bruker Cellular Analysis has always been continuously innovating on the Beacon platform and to continue to deliver new value to our users. We remain committed to continuing innovation now and into the future to further build toward a total solution to any antibody discovery need. And with that, we'll now switch over to Twist's highly anticipated presentation, where we have Colby Saunders sharing his team's amazing work to introduce Colby.
Oh, sorry, Vincent. I think it cut you off. But yeah, thank you very much. I appreciate the chance to speak with everyone on this, and thank you very much for the invite to share during this webinar series. So we're very excited to show the breadth of data that we've been able to generate on the Beacon platform and some very interesting results that we've seen within those trends across multiple different mouse strains. So not just our proprietary Diversimab and Divergemab mouse strains, but also humanized mice. So anyway, sorry, at Twist, just mentioning how our flagship is to be able to write DNA extremely quickly. So we can do this at very high scale and very high throughput. And so we can leverage that in a number of different ways, including making gene fragments, making express genes for clonal delivery, as well as variant libraries. We can also use this to support any other antibody discovery applications. And so in one way, we'll do that on the left here, and we'll focus on that in our in vivo discovery process within this webinar. And so we can do beacon discovery on multiple different species, mice, rabbits, and alpacas. We'll focus mostly on mice during this webinar to highlight the different Diversimab and Divergemab, as well as humanized mouse platforms that we're able to support. So the first that I mentioned was our hyperimmune platform with our Diversimab and Divergemab mice. Now, these are important to highlight because for very difficult targets where you have very high homology, these mice are extremely potent in being able to find binders to even low immunogenic epitopes. And so this can give not only broad diversity, but also generate hits to even self-targets. We see on the bottom left here, even a 100% high homology, a self-target, is able to mount a very potent immune response in our Diversimab mice, whereas in a wild-type mouse, in this case showing a BALB-C, there is no immune response in these high homology targets. And so this leads obviously to very nice cross-reactive antibodies, not only for human cyno, but again, for human murine cross-reactive clones, where we're able to see a number of co-localized hits to both human and murine target. As I mentioned, this also produces a wide breadth of epitopic diversity. So here we're showing against one target that's very large and typically can have immunodominant epitopes. With the Diversimab and Divergemab mice, we see dozens of different epitopes that are targeted when immunized with this particular protein. And so we have that wide epitopic coverage that enables you to find the exact epitope of interest that may be the most functional or the most specific for your particular application. Now, this also produces high affinity clones, even within a very rapid 18-day immunization schedule. Now, this owes to the uniqueness of the murine model and the genetic mutations that have been made. But as you can see here across, in this case, just showing 66 different projects, almost all of them can find single-digit or sub-nanomolar clones within each of these projects, particularly in monovalent affinity format. So this even rapid schedule is still able to produce these very high affinity hits. More importantly, even, is the broad diversity of affinity that we can find. So not only do we find epitopic diversity, but broad affinity diversity, which can be very important for particular applications such as, say, ADCs, CAR T therapy, or other technologies that may suffer from off-target toxicity if the affinity is too high. Now, Vincent already gave an excellent overview for this particular use case and single B-cell screening on the Beacon, so I won't go into great detail here. But again, we can utilize mice, rabbits, or alpacas on the platform, and we perform that sequential multiplexed assay format that Vincent was describing, and then export hits for downstream sequencing and subsequent recombinant expression. This is one example of a project where we can fully utilize that multiplex screening. So very typically, On average, we're running about 4 different independent sequential assays on the beacon. In this case, we're showing in the first assay not only protein binding, but also a ligand blocking assay. So we can measure if an antibody is able to disrupt a natural receptor ligand interaction. Then in the second assay, we're measuring binding to the endogenous target that's expressed on the cell surface. Then we're also assessing for cyno and murine cross-reactivity in addition to the human reactivity that we were assessing in the first and second assays. And then even in assay 4, now we're looking at benchmark antibody binding. So if we have a positive control antibody that we know binds an epitope that we're interested in or maybe not interested in, then we're able to look for similar binding to that particular epitope. Now, in this example, we can see about 274 hits that were of interest, but more importantly, we can down select that to the 47 hits that were of most important, meaning they were cross-reactive, on-cell binders, and ligand blockers. We can obviously supplement that with other hit profiles, but this allows us to then select the top hits for downstream recombinant expression. Now, in this particular use case, we're moving into a very tough target. So here we are targeting a proteoglycan, and more specifically, the glycan on that proteoglycan. So a very small, very specific epitope. So on the left, we can see that we're able to elicit a nice immune response in our diversimab mice, even to that very specific glycan epitope. On the beacon screening, we're able to screen, again, in the sequential assay formats to look for target-specific binding. In this case, we screened over 65,000 single B cells. We were able to yield 245 target binding hits, which is right around 0.4% of all B cells screened, which is very near the middle of the positive range that we typically would see, which is about 0.1 to 1%. After pulling the lead hits forward and humanizing them, we were able to see different activity in vitro. In this particular use case, the idea was to induce cytotoxicity into various different cancer cell lines, including some very difficult neuroblastoma, ovarian, and pancreatic lines. This was then converted into a more potent ADC and measured in various in vivo models, in this case showing a prostate cancer model where we saw complete regression of the tumor, even at a lower 3 mg per kg dosage, which is very impressive for this to show that not only was this excellent target selection on the partner's behalf, but also owed to our wonderful technology to be able to find very highly potent hits binding very specifically to that glycan on the proteoglycan and able to target and outcompete the standard of care within this assay. Now, zooming out and looking at it outside of just case study perspectives and saying, well, how does this technology perform on the whole? On the left, you'll see, on average, we still do see both hybridoma and single B cell screening workflows. And so interesting that both are still very much a key part of our discovery process. Now, zooming into some of those more difficult cell surface targets, and in particular about the 1/4 of targets that we see, which we would classify as very difficult, meaning multi-pass proteins, either in the form of GPCRs, ion channels, other very small targets like carbohydrates. About 1/4 of those cell surface targets fall into that category. And of those, what we typically will see is about 78 hits. So right around 100 hits per project for an average sized project. And the majority of those fall, greater than 75% of them, fall into the dozens of hits for the particular project. So again, even with the most difficult targets, we're still able to pull forward very valuable hits for any of these particular campaigns. Now, again, looking at them in a little bit more detail and comparing beacon single B cell discovery to hybridoma discovery, we see on average more hits from a beacon-based campaign than a hybridoma campaign. We are able to still perform high throughput flow cytometry for these hybridoma campaigns to support that modality. But again, it's very interesting to then at the top also see the differential between what a partner typically comes to us with for a project and which route they go, either for a single B cell project or a hybridoma project. Now, one thing that we note are more cell-based projects in general on the beacon. There's still a very high prevalent and very strong trend towards single pass transmembrane proteins. So I think one belief is that more and more companies are working toward difficult multi-pass transmembrane proteins. But I think one thing that we've observed is that's not always the case. There are still very novel and a lot of interest in the single pass transmembrane proteins as well. While we see a large number of campaigns and hybridoma projects to these single pass transmembrane proteins, soluble proteins do tend to be the majority of projects that we see for a hybridoma campaign. Now, looking at the beacon campaigns in more detail, we very interestingly can map the frequency of hit, which we see on the left y-axis, to the titer, which we see on the x-axis. Now, in green on the right y-axis, we see the frequency at which we observe that titer, and we can map that, as I mentioned, to the number of hits that we see. Now, this is normalized per 10,000 cells. So again, on a typical project, if we're screening 20,000, 30,000, 40,000 cells, we can extrapolate the total number of hits from there. However, very interesting that we see for our diversimab and divergemab mice that once we achieve greater than 8,000 titer, we see this jump from about twofold, from about 50 up to about 100 binders per 10,000 cell containing pens. And so we see this trend continue as titer goes up. Most frequently, we will achieve titer of 1 to 72,000 for these campaigns prior to moving to screening. Now, we can take that in comparison to humanized mice. And in this particular case, we group humanized and as well as some wild type and knockout mouse strains together. And here we see, again, a similar trend in titer that's achieved, with the most frequent titer being 1 to 72,900. But for these particular mice, we need to achieve something above that level to see that doubling of hit count per 10,000 B cells. So again, moving from 50 to approximately 100 requires that 1 to 72,000 titer. So some very interesting trends that we've observed. More importantly, this helps us understand what the expectation is for any individual campaign that may come our way, and we can see this expectation early on during the titer test stage of that project. Now, for the particular tissue that we are then able to extract from the mice that show titer, in this case showing diversimab and divergemab mice, so our hyperimmune strains very specifically here, but this trend does hold across all mice, is that we see a much higher prevalence of hit from the lymph node tissue than from the spleen. Still a decent number of hits from the spleen, but very interesting that we see this very consistent trend. In particular, more than a twofold, almost a threefold increase in the hit count between these two different tissue types. Speaking about the serum titer and how that correlates to the different tissue, that tends to be a very consistent measurement where, again, we see doubling of the hit rate, whether that's in the lymph node or in the spleen, once we achieve that titer of 1 to 24,000 or greater. Now, from the diversimab and divergemab mice, those come out as murine antibodies. So, of course, we need to move forward and humanize those particular candidates. And we've developed internal methods for doing that. In particular, not only do we calculate a humanness score, but we also can predict a binding success score. And so we're able to do this so that we can very pointedly select the most important candidates. Here we see in the green box or in the two blue boxes what the highest priority hits would be to select during this particular workflow. So this can help us to, again, predict which binders we should move forward with and express to be very efficient about this particular workflow. And to show that these work very well downstream, not just for binding, but also for developability, we can run them through a battery of developability assays, including CIC, HIC, BVP ELISA, AC-SINs, and DLS and DSF analyses to be able to compare them to these 12 clinical standards that we show here in this particular figure that have a range of different developability scores. And you can see that all of the humanized candidates that we pulled forward fell within the excellent developability score range. Now, of course, if you don't elect to humanize, we also have the capability of running humanized mice. So these mice produce fully human antibodies from the B cells. These are engineered mice. We partner with Transchromosomics to do this. And so now I'll get into the anti-CD22 campaign that we ran in order to showcase this capability. So again, we immunized Transchromosomics mice. We had 2 different cohorts and 2 cohorts of hyperimmune mice in comparison. So we used our rapid protocol with our diversimab and divergemab mice, and we had both a rapid protocol and different extended protocols used with the Transchromosomics or TC mice. Of course, we saw excellent titer in both strains. As expected in the diversimab and divergemab mice, we saw higher titer. Again, these mice are engineered in order to enable that. And so not a big surprise there, but we were very happy to see the different titers over background for the TC mice, both to the recombinant protein version, so CD22 recombinant protein, as well as to target positive cells. In this case, we use Raji cells as the positive and Jurkat cells as the negative. Now, we also, at the same time that immunization is occurring, we perform all of our on-beacon QC. And so this can include labeling recombinant protein fluorescently to be able to enable the on-beacon detection, which you can see on the right, as well as confirming target expression on cells by flow cytometry, and also optimizing import conditions on the beacon for each of the different cell types that will be used for the particular campaign. Very important to get a nice homogeneous population on the beacon chip when doing the screening. Now, this is example outputs of this particular screen, where again, we used 4 different sequential assays. In the first assay, we used an AVID format to screen for any off-target binding. In the second assay, we used the negative cell line, so the Jurkat cells, which we did not observe binding to. In the third assay, here we're binding to the endogenous target on CD22 on Raji cells. Then in the fourth assay, we're able to see the on-protein binding profile, CD22, which was also multiplexed with IgG secretion to confirm binding in that format. What we see when we compare the different outputs, on the left, we see IgG secretion. Clearly, this is an important measure because we would not be able to detect binding if we didn't have IgG secretion. Again, the diversimab and divergemab mice are engineered to have more productive B cells, and so we did see that trend continue, but we were able to see very good output from the Transchromosomics mice. Again, more prevalent in the rapid immunization scheme than in the extended, either 5 or 8 week extended immunization schemes. But interestingly, the hit rate is higher on a per IgG secreting B cell rate for the extended campaigns. And so we're able to see a large hit rate from both rapid or extended immunization schemes for the TC mice. When looking at the final output, we do see a differential in diversity of the unique VH and VL paired sequences. So again, as expected, hyperimmune mice are producing more diverse sequences by design due to the genetic engineering, but we were able to see a 62% unique rate for the Transchromosomics mice, which falls very much in line with what we've observed from other humanized mice, as well as wild type and knockout mice that are generally between the 60 to 65% unique rate. Looking at that in a bit more detail, we can see on the left, the hyperimmune mice produce a very wide variety of different V gene and VDJ combinations. On the top is the heavy chain, the bottom is the light chain. And on the x-axis are all the different V genes that are used, and the y-axis are the different J genes that are used. And so again, we can see a very broad and wide range of different combinations, and this leads to very few clonal lineage clusters, which we see on the bottom. In blue, that means it's an identical clone, and the further red, the more divergent the sequence is from one another in this particular heat map. On the right, in the transchromosomic humanized mice, we still see very good diversity output when comparing to other humanized or wild type mice. We see broad V gene usage across multiple different V gene families, including 1, 2, 3, and 5. Again, pairing with different J genes to give us nice, unique, heavy CDR3s. Same with the light chain, where we see broad light chain V gene usage and VJ combination. We do, of course, see a dominant clonal lineage. Again, this is expected in non-hyperimmune mice because they typically can focus on a dominant, immunodominant epitope and would expand a cluster out based off of that particular epitope. And so we are able to observe that happening in a portion of the B cells that were derived from this particular campaign. Now, looking at the output and downstream characterization, we're able to see a few different very interesting trends. So as expected in terms of output affinity, from the rapid immunization scheme, we see a nice range of single from double-digit and into single-digit nanomolar affinity clones. Of course, from extended immunization schemes in these mice, we're able to see a higher prevalence of single-digit and then extending down into sub-nanomolar clones. This is due to the increased amount of somatic hypermutation. Now, when we look at Beacon single B cell affinity ranking, so this is a feature that we haven't mentioned yet, but one of the abilities that we can do on the Beacon when we multiplex IgG secretion with recombinant protein target binding, we're able to derive a relative affinity score from that single B cell screening process. And when we correlate that, which is shown on the y-axis, to our downstream affinity by SPR on the x-axis, we can see a pretty nice correlation. Of course, there are a couple of outliers, but considering we can get this y-axis measurement on the hundreds or thousands of hits that we are seeing on the Beacon, this can produce a nice screening criteria that we're able to bias towards when we make our downstream selections. Now, on the right, this is highlighting another feature that we particularly like by utilizing on-cell screening on the Beacon. Now, this is showing in blue clones that were selected based off of protein-based screening only. So we did not incorporate cell-based screening for these particular hits in order to assess this comparison. Now, on the x-axis, we see the downstream validation on our target-positive cells, the Raji cells. And on the y-axis, we see ELISA binding to our recombinant protein, CD22. So, of course, the majority of those are able to bind to the protein, but it's interesting that we see a smaller portion that are able to bind to the cells and the target, the endogenously expressed target on cells, probably because it's binding an epitope that may not be folded exactly the same in the recombinant-based version of the protein and the cell-based version. But interestingly, in red, we see a much higher portion of on-cell binding validated clones because these clones in red were screened on the Beacon for on-cell binding. And we can also see this additional population that are binding an epitope that is present on cells that is not present on the recombinant protein. So again, very important to incorporate both aspects, both protein binding if a recombinant protein is available for a protein like a single pass transmembrane protein such as CD22, so that we can draw some of these more in-depth affinity correlations, but equally important to screen on cells so that we ensure we're also pulling forward relevant epitopes. So with that, I will end and we can move to the question and answer session. Please feel free certainly to reach out if you'd like to learn more about any of these campaigns or campaigns that we could run in conjunction with you. And I thank you for your time.
Yeah, so we can get started in the Q&A. So thanks everybody for submitting questions. Feel free to continue to submit these. I'll post those as they come in. But we received the first question. Colby, this is to you. What drives the decision for single B cell versus hybridoma most? From your perspective, would it be something related to budget or epitope space or diversity or something else?
Yes, great question. So a few different factors there. So one certainly would be the ability to run high diversity campaigns. So in other words, if someone would really like to explore full epitopic diversity of any particular target, then they would be able to run both the diversimab and divergemab mice and the humanized mice. And that would be a good idea for those particular campaigns, as well as any very high homology target campaigns, as mentioned at the beginning. Now, we can see some very good results from the humanized mice and very rapid responses and able to generate a fast, fully human antibody. And so we like to run both in parallel so that we can not only get the epitopic diversity that's important from the diversimab and divergemab mice, but also be able to sprint to the fastest human clone with the humanized mice.
Great. Another question for you, Colby. Have you compared bone marrow samples to kind of your lymph node and spleen samples, and kind of what results you would get from those?
Bone marrow, lymph, and spleen. And one thing that we have pointedly noticed is that for bone marrow samples, the key is that we see the highest productivity if you extend the immunization out as long as possible. So we've typically moved to bone marrow if the titer is low during initial immunization processes, and we then have extended out to very long immunization schemes. But certainly in the short-term immunization schemes, lymph node and spleen are the more productive tissues.
All right. Another question for you. My understanding is that in general, you generate higher affinity antibodies by extending your immunization scheme out to 8 weeks. Does the overall number of clonotypes and the diversity that you get stay similar compared to that rapid protocol, or do you see a change in diversity? We saw that, I think, in the CD22 case study that you presented.
Right. Yes. No, that's a good question. So certainly in the diversimab and divergemab, we see good clonal diversity across all time points. So even if we need to extend out, again, that is a byproduct of the genetic engineering and a pointed feature that we've built into those mice. For other mice, for humanized mice, as well as wild type and knockout animals, we have noticed that there is a clonal contraction over time. And so the longer you immunize, the more the clonal contraction does occur. However, we are able to adjust immunization schemes, and we are able to also take additional mice. So I think the other important feature to note is that by taking multiple mice to screening at various time points, we can build in additional diversity by doing that, because any 2 mice or 2 animals will develop very different responses than one another over time. I'm sorry, it looks like my camera is not working, so apologies.
That's all right. And this is, I guess, the final question that I can see right now, but this is actually my own. Yeah, just generally, when would you choose between using the diversimab versus the divergemab versus this specific TC mouse or even any other transgenic mouse model that Twist might be thinking about? How might you kind of paint that landscape of how you choose a particular model system to go forward?
Yes, yeah, no, very, very good question. And so I think one thing that we think about from a scientific perspective, again, is broad diversity. And so we want to be able to use both in conjunction, divergimab, and humanized mice to be able to afford the largest diversity possible, but also commercial terms as well. So we know it's important for our partners to be able to have flexible commercial terms. And so we've been able to select humanized mice to use that will enable us with that largest flexibility possible there.
All right. I don't see any other questions. So I think what we'll do is end this webinar. Thank you so much, Colby, for your time, and thank you to the audience for taking the time to watch and have us present. But yeah, we have 2 more webinar series, seminars or webinars coming up in the rest of our series. So stay tuned. We have the next one on April 10th, and then followed, Colby and I will be back to have a roundtable discussion on 16th. Thank you, everyone, and take care.
Awesome. Thanks, Vincent.
Mm-hmm.